Hello, I am Fei Betsu and I'm the director of the Biological Resource Center of Institut Pasteur. The Biological Resources Center of Institut Pasteur is transversal biobank infrastructure, including collections of specimens of human origin and different collections of microbial strains. And these collections are either historical or prospective. Our activities are the typical activities of a biobank, and these include the reception, processing, quality control, preservation, and distribution of specimens and associated data in compliance, of course, with applicable national and international regulations and safety standards. Our mission is to support impactful and reproducible research by the Institut Pasteur teams and researchers in general by making available qualified microbial and human origin bioresources. Qualified means fit for the intended purpose. Apart from enabling access to bacteria, cyanobacteria, or human samples, we also accept the deposits of bacterial strains from depositors from all over the world. Our processing methods include live cultures, lyophilization, molecular extractions, or cellular isolations. While our characterization methods include methods for genomic, phenotypic, but also functional characterization of the different types of biospecimens. I would also like to say that characterization includes methods for quality control in terms of authentication, viability, or integrity, and methods for high added value annotations in terms of antibiotic resistance or microbiome profiling, for example. You will now hear more from Muriel Guger, Dominique Clermont, Emmanuel Roux, and Raquel Hurtado Ortiz on the specific units of the CRBIP.
Hello, I am Muriel Guger. I'm the head of the collection of the cyanobacteria at the Institut Pasteur and at the University Paris-Cité. The laboratory is maintaining the collection named Pasteur Cultures of Cyanobacteria, or PCC. This collection represents the diversity of physiology and ecology of this phylum. The PCC is a collection of living pure organisms which are monoclonal strain. They are maintained by perpetual transfer under controlled environmental conditions since 1971, and today we have 800 strains. The collection comprises a large number of genera, species isolated from a wide variety of habitats. The strains are conserved either in solid media, like I show you in the picture at the top, or in liquid media, like I show you in the picture in the middle, in accordance with the origin of the organisms. The strains are also cryopreserved, but the revival of a strain takes between 1 to 4 months. Thus, we prefer to keep the strain alive. The cyanobacteria are known to produce cyanotoxins. Those toxins are hepatotoxin, neurotoxin, dermatotoxin, and cytotoxin. Thus, it represents a hazard for humans and animals, notably through drinking water or in recreational areas. But moreover, the cyanobacteria are capable to produce a diversity of natural products. More than 80% of their biosynthetic gene clusters that we can recognize in their genome are not associated with any compounds, while there's more than 1,500 chemical structures which have been described. I show you on the last picture the compounds which are produced by Microcystis and Planktothrix, strain that represent toxic bloom in the freshwater environment. And you can see that we still have several orphan gene clusters that we can't associate to any compounds. Finally, I'll show you two examples of 10 years of collaboration with chemists from all over the world to discover new compounds and novel chemistry. Notably, the investigation of the natural product pathway of the PCC strains led to the discovery of 20 new natural products, but also novel enzymes and a novel chemistry, like in the Morinaka et al. That I showed you on the side. Finally, the last example is the natural recombinatorial modularity of the trans-AT polyketase to actin disrupting polyketide. Understanding the biosynthesis of the polyketide family, which inhibits the polymerization of actin, conducted to the study of this compound acting on tunneling nanotube formation, notably to stop the propagation of neuronal disease through this actin-containing membrane protrusion. And here I suggest that you can look at two publications that discovered these compounds and also their bioactivity. Thank you.
Hello, I am Dominique Clermont, head of the collection of Institut Pasteur, the CIP, from Institut Pasteur, Université Paris-Cité. Bacteria of lineages other than the cyanobacteria are preserved at the CIP. It was Émile Roux and Alexandre Yersin who first had the idea to conserve a set of bacteria for practical courses of microbiology at Institut Pasteur. Then Jean Benoît, really the father of the CIP, had the ambition to conserve and distribute all known microbes. René Legroux and Louis II, on their side, developed a gelatin maceration medium which make it possible to maintain the microbes for a long time, up to 15 years in the cooler. Over the following years, freezing and freeze-drying were routinely used to preserve bacteria over time. With Michel Piéchaud, the CIP became a reference center recognized nationally and internationally. In 1991, Chantal Bizet was appointed head of the CIP. She implemented the quality management system, and the CIP was certified for the first time in 1998, according to the ISO 9001 standard. She also created the Biological Resource Center of Institut Pasteur, the CRBIP, in 2001. At the present, the CIP includes a wide biodiversity of bacterial strains isolated all over the world, from human, animal, food, plant, and environment. These bacteria are distributed for education, quality control, and research. Moreover, the research activity of the CIP are focused on taxonomy studies with a description of new bacterial species, on the discovery of natural products in collaboration with other scientific teams, and the setting of a collection of anaerobic bacteria isolated from stools collected by CRBIP Biobank. Thank you.
My name is Emmanuel Roux. I am the head of the CRBIP Biobank unit. The purpose of this unit is to ensure the management of human biospecimens collected either from healthy volunteers or from patients, in the context of specific clinical trials, but also in the context of open collections. We then speak of collections of human samples. Our unit ensures the control and documentation of critical pre-analytical parameters, in particular in the context of our activities of reception, preparation, characterization, and preservation of these human samples. Our main mission is to provide samples that are fit for purpose to researchers, who then can produce reliable research results. We work in close collaboration with medical departments of the Institut Pasteur, as well as with the clinical investigation structures that operate medical consultation and can ensure specimen collection and appropriate donor or patient consenting. The specimens we receive are of very diverse types: whole blood, swab, stool, urine, and many more. From these specimens, simple derivatives can be produced, such as serum or plasma, but also more complex molecular derivatives such as DNA or RNA, and cellular derivatives such as blood monocytes. Last but not least, some types of specimens can be used for downstream isolation for microbial strains. In this case, we talk about microbial derivatives. The specimens are then collected, processed, and stored in such a way as to best preserve the viable human microbiome. For example, stool can be transported and processed in anaerobic conditions before freezing. On the other hand, human samples are used for research on disease caused by microorganisms, or research on compounds produced by microorganisms, and that can be used to cure human diseases. For example, serum samples are needed for the validation of new diagnostic biomarkers. Human cell lines are used to validate new bioactive compounds, such as protease inhibitors. Blood mononuclear cells are needed to study immunity mechanisms against pathogens. Sometimes, such research may lead to an invention and patent applications. Thank you for your attention.
Once a discovery based on the utilization of a new bioactive compound or of a new cell line is made, the inventors can deposit the material at collections recognized for purposes of patent procedures. Hello, my name is Raquel Hurtado Ortiz. I work at the Biological Resource Center of the Institut Pasteur, and I am the head of the National Collection of Cultures of Microorganisms, also known as the CNCM. The CNCM hosts all types of biological resources, both microbial and human origin resources, for purposes of patent procedure. This biobank exists since 1976, and since 1984, it has been recognized as International Depositary Authority under the WIPO-administrated Budapest Treaty on the international recognition of the deposit of microorganisms for the purposes of patent procedure. Since then, it is the only international depositary authority in France. Nowadays, there are 48 international depositary authorities in operation around the world, and biological material deposit in any of them is recognized by all members of the treaty as valid for patent purposes in all countries where protection is sought for a specific invention. We have a great diversity of biological resources, from microbiological resources such as bacteria, virus, filamentous fungi, and yeasts, to hybridoma cells or human and animal cell lines. The importance of the CNCM lies in the fact that when using microorganisms for patent procedures, the written disclosure must be complemented by the deposit of the biological material in collections recognized for this purpose. Moreover, the Budapest Treaty ensures the availability of microorganisms to reproduce invention-related findings and eliminates the need to deposit multiple samples of the same biological material with biobanks in different countries. Patent applications are increasing year by year, and for this reason, the biobanks and collections must be at the forefront to ensure the conservation and availability of these precious biological materials.